Good morning, ninth grade. We are going to be creating or making our own animal cell. And inside the animal cell, or because we're doing it ourselves on top of the animal cell, we will be placing the organelles. The purpose or the objective of this assignment is to allow you to touch and feel and manipulate the organelles. Take a look at the shape of the organelles on a diagram. Find a place to place it on the cell, because you need to know the activeness of the organelles that are on the cell. You need to know the structure. You need to know the function. In other words, what role do these cells play in a cell? And you need to know that they interact together. Those are the standards, and they are the objective of this assignment. So I want you to start, because I put this thing together completely as an example. I want you to start simply with the cell, just a cell. Make a circle, cut it out of paper, draw it, however you'd like to do it. Make a cell, and then for me, this little blue thing in the middle is the nucleus. Now, on top of the nucleus, I want these little squiggly lines. I don't care if you draw them on or you use paper and cut out squiggly lines, however you choose to do it. That represents the DNA, the genetic material in a cell, because the nucleus is the brain of the cell. It is the blueprint. And then I want you to make a round structure here that represents the nucleolus. If all you have is a cell, the nucleus, the chromatin, and the nucleolus, then you already have 5 of the 12 or 13 organelles we're going to put on this cell. I'm going to test you on 12, just an assessment. But you may as well go ahead and place 13, because they all work together. Now, what are the 5 organelles? Well, the first is the outside of the cell. This is an animal cell. It's the plasma membrane. Typically, the animal cell is shaped like a circle. And the plasma membrane, what I did was I pointed to the edge of the cell, lined right to the edge. I put a tag, plasma membrane. Inside the tag, I wrote the function of the plasma membrane, which is to separate the inside of the cell from the outside of the cell. The second organelle is the nucleus, because in a cell you have 2 main organelles. You have the nucleus and you have the cytoplasm. This entire blue thing in the middle is the nucleus. The area around it is the cytoplasm. All of the organelles sit on top of the cytoplasm. And so the cytoplasm is a jelly-like substance that hold the organelles sort of in place. I mean, they're not stiff because they do move. Things move in the cell, but it keeps them, kind of stable. So if we were using liquid materials, you could use Vaseline and stick organelles in Vaseline, and that Vaseline would be the cytoplasm. So there's plasma membrane, nucleus, the cytoplasm, the DNA material, and the nucleolus. And then I want you to pause this video. And I want you to get that done. And I want you to label it. Now, when you come back, I want you to add, let's say, 4 more organelles. This right here is the endoplasmic reticulum, this whole thing. But some of them, half of it has dots on it. Those dots represent ribosomes. The nucleolus makes ribosomes, and ribosomes make protein. And ribosomes are easy to distinguish on the cell because they're usually the smallest little dots. Even if you have a comprehensive cell, the smallest dots scattered all over the cell, and then on top of one of the organs, which is typically the rough endoplasmic reticulum, that's the ribosomes. And the smooth endoplasmic reticulum looks like the rough, but it does not have any ribosomes attached. And then you've got smooth and rough endoplasmic reticulum, and then you have Golgi apparatus. Now, there's a reason for why I put the smooth and rough endoplasmic reticulum, the ribosomes, and the Golgi apparatus on next. And that's because when ribosomes are made in the nucleolus and they move out of the nucleus, because they're going to move away from the nucleolus, they're going to move out of the nucleus, some of them will attach to the ribosome and some will be free floating in the cytoplasm. But the role of the ribosome is to make protein. And so protein, once it's made by the ribosome, it's stored in the endoplasmic reticulum, the rough one. So the organ receives the protein from the ribosomes that are sitting on top of it, and likely from some that are floating in the cell itself, floating in the cytoplasm, and it receives the protein, packages it, because the protein will be shipped in little vesicles to the endoplasmic reticulum. And the endoplasmic reticulum receives the protein and then addresses it like it's a post office or virtually like an email. This is technology, and presses send. From the Golgi apparatus, that protein now goes to where it's needed, to wherever it's needed, essentially. Sometimes it's right outside the cell. So when you see the folded material that's closer to the edge of the cell, likely it's Golgi apparatus, because both of these organelles, or all three of them, have folds, and they look similar when you see a picture of them on a cell. But if there's one that's closer to the nucleus, or two of them that are closer to the nucleus, that's likely the endoplasmic reticulum, rough and smooth. Just look for the ribosomes. And one that's further into closer to the plasma membrane or cell membrane might be Golgi apparatus. That's just a rule of thumb, and it might help you for placement too. So those four organelles: ribosomes, rough endoplasmic reticulum, smooth. What does the smooth endoplasmic reticulum do? It stores the lipids. And remember, lipids are fats, so it stores the lipids. And of course, ribosomes make protein. Pause the video. I want you to place the organelles on your cell, and I want you to label them. On the outside, I want you to put, like here is chromatin, point it directly to chromatin. Plasma membrane, point it directly to plasma membrane. Rough endoplasmic reticulum, right here. So point directly to the organelle. And with the paper, write the name on the outside. Because when you study, you want to be able to identify these, and you want to be able to identify their roles. And if you slip up or you forget, you just open the sheet, and it'll give you a reminder until you no longer need to open it anymore. Pause the video, get that done, and then we'll close this out. Well, you're back. Now we're going to finish this out with 1, 2, 3, 4 more organelles. We don't survive unless we have energy. We get our food from energy. And our food is broken down and converted, of course, for us, protein, lipids, and carbohydrates. The energy in a cell, animal cell, is processed by the mitochondria. And it's turned into ATP. That's the energy used by the animal cell, ATP. And so it's kind of shaped like a peanut or like a kidney or like a kidney bean. I don't know if you guys have seen kidney beans, but that's what this is shaped like. And so it's easy to recognize it. Sometimes it looks like a little cup when you take a look at it on the cell, and it always has these little waves or little folds inside. That's energy. The next organelle is lysosome. Lysosomes break down things that the cell doesn't need anymore. It's virtually digested. It has digestive enzymes. So when we eat, our food is digested in the stomach. Well, actually, digestion begins in the mouth, but that's another thing. But it's digestion, it takes place in the stomach. Lysosomes are responsible for digestion. And here, lysosomes, and as you can see, I have it pointed right to the lysosome card. The other two organelles, when you see lines, just straight lines going through a cell, that's cytoskeleton. And all that does is give the cell its structure, give it some structure. Centrioles, which when we get to, we're going to get to centrioles in details when we're back up into DNA and chromosomes, etc. But on the cell, centrioles really remind me of licorice, Twizzlers. And that's the reason why I put two little Twizzlers up here, because centrioles also help with the structure. So once these are all labeled, work at trying to recognize by shape and identifying the parts. You've seen videos on it. I've submitted videos. You've had hard copy of it. And now you're actually doing it. And then when you can flip, or when you no longer need to flip up the card, you know that mitochondria is for energy. Where are the ribosomes made? They're made in a nucleolus. What do ribosomes do? They make protein. Where do they take the protein? They take it right to the rough endoplasmic reticulum. Oh, really? Well, where does it go from there? To the Golgi apparatus. And then the Golgi apparatus takes it to where it needs to go, or sends it to where it needs to go. If you have any questions whatsoever, just give me a call. But remember, the objective is to help you to recognize and know the structure of the organelles in this cell, to understand their functions. In other words, what do they do in the cell? And to be able to understand the interrelatedness, digestion, storage, energy. And then in another lesson, we'll talk about what happens as things leave the cell. Have a great day. Miss Champion is out.